Well, welcome as we explore the fantastic world of the cerebellum. My name is Doug Major, and I have no disclaimers or disclosures. Leonardo da Vinci, while making wax casting as he tried to understand anatomy, found this small area in the back of the human brain that he called the cerebellum, just a small hemisphere in the back, and that's where it came his name. Purkinje, one of the first to stain the back of the brain, found these elaborate and beautiful tree-like structures. And of course, he grabbed that for himself as the Purkinje fibers. Cajal follows up, does this wonderful drawings, finds out that there is a space between the synapses, and that brings us to today. In my area, interest in corrections, Lombroso, looking at a serial killer, dissecting him, finding that, hey, he had a smaller cerebellum, that there was something wrong with his brain, the biological theories of criminality. And we really appreciate the work of Dr. Yang and Hanu, that I'm hoping you all listen to him. We have this great follow-up now. They did the academics, and now we're going to do hopefully some applied. Start with a priest test that I was taking in my Z Health training. See how you do with these. Your new VT patients complain about a muscular strain around his eyes while performing smooth pursuits diagonally and down to the left. You decide to activate the associated ocular muscles with a reflexive eye movement instead of a voluntary one. Which of the following therapies would best accomplish this goal? An angular VOR drill for the patient's right posterior semicircular canal, an angular drill for the patient's right horizontal semicircular canal, a VOR cancellation drill for the patient's left posterior semicircular canal, a gaze fixation drill with the patients diagonally down and to the left. Not too easy. Second question, your patient's a boxer having a hard time keeping her eyes stable in her opponent when she dips down during sparring. Which of the following drills will most strongly affect the cerebellar structure responsible for optimizing her upward eye movements as she quickly squats down? An unpredictable horizontal smooth pursuit performed sinusoidally. A fast A VOR drill with the posterior semicircular canals followed by an upward smooth pursuit. Vertical saccades performed in time with a metronome. A slow A VOR drill with anterior semicircular canals followed by a downward smooth pursuit. These are all in your handouts, by the way. Got it going. Get going fast. After observing your patient is walking with his left shoulder and his left hip internally rotated each side, to use some motor stimulation stacked with oculomotor input to help provide corrective activation for this neurological gait pattern. Which of the following combinations would be most stimulating for the brain structure of interest? Complex nonlinear body movements on her left side while fixating on a target in an eye left position. Complex body movements on her left side while fixating a target in her right eye position. Complex nonlinear body movements with the right side while fixating a target on her left position. And simpler uniplanar body movements on her left side while fixating a target on the right position. Sort of hard. Did you notice that wasn't easy? I was surprised how difficult it was. So I found a gap in my knowledge. Today we're going to mind the gap. Let's fill the gap. First of all, let's do some testing of the cerebellum. Hands up, everybody, please participate. We're going to go ahead and feel that first knuckle, and we're going to tap as fast as we can. Ready? Let's begin. Go, 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 go. Keep going. Keep going. Keep going. If you notice a slight difference between the two hands, that's sort of normal with your dominant. But if one's a lot worse, indication that maybe a cerebellar weakness on that side. So why the cerebellum? Well, we used to just call it a motor system, but now it's connected with autonomics, the posture system, the oral system, the ocular motor system, the vergent system, and the higher cognition areas. And so at this time, I'd like to introduce to Reza Shadmir. He's got a great lab at Johns Hopkins, and Johns Hopkins is a hotspot for cerebellar work. He's put together a wonderful webinar, and we're going to hear from him.
We use engineering to describe very complicated systems, satellites, large ecosystems. Why don't we use the same tools to try to understand the brain? When I pick it up, your hand moves up a little bit, but when you pick it up, it remains absolutely steady. When you do something, your brain predicts what's going to be the consequences of it. And that prediction is a computation that is performing. Through practice, it's learned physics. What we just saw was that the brain predicts things, and based on its prediction, it can maintain control. We've learned over the years that this comes from a specific part of the brain, the cerebellum. For every movement we make, we need the cerebellum. I really had two problems to solve. The first problem was, when you just normally move, what is the cerebellum doing? But then when you learn to do something new, how does it add to it? And to do that, we had to study neurons in the brain. We have been able to describe a model with which we can understand certain properties of neurons in the cerebellum. And we remain hopeful that by allowing us to use the model to decode the activity, we've taken the first step in understanding how learning takes place. Diseases that affect the brain result in almost always some form of a movement disorder. And when there's damage to the cerebellum, people really struggle in ordinary things. The implication of our work is that it's possible to make headway in understanding how did this come about. And that's really the promise of neuroengineering, is that if you understand the system well enough, you can diagnose simply by watching people the specific location where something has gone wrong. And then perhaps if you know how that part functions, you would also know what should we do to repair it.
Yeah, so what should we do to repair it? What got me really excited about the cerebellum was in Chicago at Academy meeting. Tara Alvarez, working with Mitch Scheiman, was talking about the neurology of the CITT. And she presented this, and I just stood up in my chair and said, wow, everybody else quiet. I'm so excited. And she talked about the oculomotor vermis. Well, I knew all the Brodmann's area. What about this vermis? And the oculomotor vermis was so important. It was the place where most of the learning took place and the biggest change after vision therapy. But I didn't know, and that's part of my quest that I'm sharing with you today. And of course, I'm so thankful that Tara also can make a comment for us today. Tara Alvarez.
Hi, my name is Tara Alvarez. I'm a distinguished professor at the New Jersey Institute of Technology in biomedical engineering. And you may wonder why an engineer is interested in vision therapy for convergence insufficiency. But to me, it's an ideal model to study neuroplasticity in the brain. Specifically, we are looking at how the brain reorganizes in terms of synchronization, recruitment, and connectivity, where the ocular motor vermis in the cerebellum is the primary region that is responsible for the changes that we see in terms of improvements in patients and how it talks to the frontal eye fields, the supplementary eye fields, and the parietal eye fields. And I look forward to learning more and sharing it with all of you.
And Tara is going to be sharing that with at the next Academy meeting with a workshop. Hopefully we can be there and if you want to learn more. And of course, this is not new. Skeffington talked about the anti-gravity circle, talked about a girl walking on the train tracks with balance and movement that changed her binocularity. And remember, the cerebellum has 70 billion neurons, 4 times of that of the neocortex. And even though we always think of the neocortex as that unique human thing, the cerebellum expanded 6 times faster during evolution. Let's hear some more from Jorn.
Start the talk, the Cerebellar talk, with showing this image here. So this is an image we took of a postmortem cerebellum at 150 micron resolution. And I'm showing this just because it allows you to really appreciate how big and how intensely folded this structure has become if you compare it to a mouse or even a monkey cerebellum. Okay, it's really highly packed. And based on this scan, Marty Serino and I built the first full surface reconstruction of a human cerebellum that really lets you appreciate how intensely folded this is. And Marty spent about half a year in trying to flatten this thing out into a flat map. And this was the result. So it's a sheet of cortex that is about 1.2 meters long and about 15 centimeters wide. Okay. And it has this very interesting foliated structure here running medial to lateral. So it's folded like an accordion, basically in one direction. Now, we found that the surface area of the cerebellum was about 80% of the entire neocortex. So it's bigger than a single cerebral hemisphere. We know that it, of course, has more than half of the neurons of the human brain. But more importantly, I think evolutionary is that even though it only has 1/7 of the volume of the human brain, it consumes 1/5 of the brain metabolism. So it's actually quite a costly structure. Despite that, it has expanded in human evolution, and this is mostly in recent human evolution from the common ancestors with the Neanderthal. That's the brain structure that has expanded more than any structure in the neocortex. So there is something about the cerebellum that is intrinsically important of making or shaping human cognition. And the question, of course, that we're all interested in is, what is this? What are those processes that the cerebellum contributes to make us human?
So I think that was just right on point. And the cerebellum influences, visual processing. And for us, this was a slide shared with me at the Brain Mapping Institute by a physical therapist, actually. And the issue about how when there's impairments with the cerebellum, we make errors in velocity of moving things and sensitivity to biological motion. But the main thing I want to point out is there's no direct link with the cerebellum and the primary cortex, that the dorsal stream is we're connected to the dorsal stream, but not the ventral stream. And aren't we dorsal stream doctors? We are the vision care providers that use this processing that in the cerebellum. I had to include some comments from Jam, who did such a great job in our meeting a few years ago and is still such an advocate for us in the brain trauma area.
So here's a chart of your brain development, conception, here's birth. And so most of brain development happens here. There's neural migration, and a lot of connections are being made. The only thing that goes on all the way through adolescence is called myelination, which is basically putting a coating over the nerve axons and making them conduct faster. But there's one organ that doesn't do this, and it's the cerebellum. It develops in the first few years. So it's very different from the rest of the brain. In fact, if you look at brain growth over the first couple of years, it's really dramatic. Huge. The blue is boys, and the red is girls. Of course, girls exceed boys. Oh, that's another story. Okay, so the brain is growing here. It grows very rapidly, but the cerebellum has an 88% increase in the first year. So the tremendous increase in cerebellar size in the first year of life. In fact, if you look at cerebellar development in man, I'll just read this, but in ancestral hominids, what happened was the frontal lobes expanded dramatically. But in recent development, the increase in the relative size of the cerebellum as the neocortex reduced its size. So basically, cerebellum has increased in size compared to the prefrontal cortex. What's interesting is what you see in, I don't compare Neanderthals and autism, but they both have very large prefrontal cortex. And in humans, late developed humans, the prefrontal cortex shrunk, the cerebellum got bigger. Why? I think because before we had good timing, we had explicit timing. You had to be always aware of somebody talking or somebody making a sound when it's going to come in. And it wasn't unconscious. The cerebellum wasn't doing it then. The cerebellum got bigger and took over those functions and took it away from the prefrontal cortex. The prefrontal cortex actually shrunk over evolution, and the cerebellum got bigger because of an unconscious function. That's why we have to talk about this now in 2014, because most people are unconscious of that function. Back in the old days, you were very conscious of it.
So as Jan was saying, more brain cells mean better prediction, and all different numbers, but 70% of all brain cells are granule cells in the cerebellum. And the idea that the cerebellum grows an impressive 88% the first year as it's learning, and how these granule cells descend and connect during that early exploration time. as we are as developmental optometrists. This is so important as they predict timing and spatial based on spatial errors. Now, I love the history. I think it's, as you get older, you really enjoy history. So Rez is going to talk about the history and right on point.
Okay, so let's begin by giving a little bit of a historical view of what I think are three main ideas that describe function of the cerebellum. The first idea begins with Gordon Holmes, a early 20th century neurologist who was examining British soldiers that were returning from World War I. And this is something that I learned from David Z. And what you notice in this picture is that the British soldier is missing the helmet that would protect the back of his head, whereas the German soldier has that helmet. So this unfortunate design flaw in the British armament led to Gordon's examination of these soldiers that had cerebellar damage. And he came up with two fundamental ideas that we still rely on to diagnose basically people with cerebellar damage. And these two ideas are the following. The first is control of movements, and that has to do with the control of the endpoint. And this led to the concept of dysmetria, the notion that the movements that these patients were making often started out okay, but then it ended poorly. The second idea was the notion of hypotonia, that postural control was also affected in these patients. They didn't produce the motor output that's required to hold still appropriately. They often got very tired voluntarily trying to hold still at a particular location, say with their arm. 30 years later, there was another wonderful and seminal idea, and this has to do with learning. This idea came from initially David Marr and James Albus, and then experimentally confirmed by Masao Ito. And this is the concept that inferior olive is the teacher of the cerebellum. The Purkinje cells are the students. So the first idea had to do with control. The second idea had to do with learning. And then the third idea, again, came 30 years later. And this was the concept that was still with us today. And this notion came from Chris Smile and Daniel Wolpert. And it is the concept of forward models. The idea that perhaps what the cerebellum is learning to do is to predict the sensory consequences of action. So you generate motor output, there's some sensory consequences associated. Perhaps this concept of forward model is what's being learned by the cerebellum. Now, I'm going to show you examples of these three ideas throughout the talk and try to put the three ideas together. But in order to do that, we need to start with a task. Now, the task that I studied for decades was a simple reaching movement that you see here. And what, a healthy individual makes these movements rather well, but a person with cerebellar degeneration begins that reach okay, but then ends up in these movements that you see here with overshooting or undershooting. And this is what Gordon Holmes had talked about with his dysmetria. Now, there are other forms of dysmetria. I'm going to show you saccades. Here's a patient attempting to make saccades. And what you're seeing is that the movement starts okay, but then it overshoots the target, overshoots. You can imagine how debilitating it would be to attempt to read when your eyes are moving in this way. It would be really difficult to really have a normal life if all movements look like this. And so it raises the fundamental question. So why does this happen? What is the cerebellum computing that's so essential for ending movements like you see in saccades? Now, to work along this direction, I need to give you a little bit background about the neurophysiology of this simple movement, this elementary movement, this saccade. So to start us out, we have information that arises directly from the retina to the superior colliculus. Essentially, the map of the visual space occurs on the superior colliculus. Superior colliculus has the machinery downstream of it that could generate an eye movement, make a saccade toward that stimulus, but it's not allowed to make that movement. Instead, the same information goes to the visual cortex and then the other parts of the cortex, frontal eye field, parietal cortex, and eventually the basal ganglia. Here, our brain evaluates what is it that we're looking at. And then if we have something of value that we would like to bring onto the fovea, then what we have is information that's sent to the colliculus, essentially giving it permission to move our eyes to the location where the stimulus of interest is located. Now, as this movement takes place, information is sent via the pons, the cerebellum, and the cerebellum makes a prediction that is going to be central for allowing this circuitry here to generate that movement well. Okay.
I hope you realize that's really right on point. The importance of that development of that concept, the hypometrae, and how that's again actually even proceeded since then. So let's talk about other areas: cerebellum and eye alignment, acquired esotropia that happens after a certain age. This is secondary not to an underlying earlier tendency and just decompensation. I would suggest all the older individuals you see with beginning esophoria and tropia, you need to check for cerebellar function. Another area, meta-analysis, just again, January, a couple months ago, PTSD associated with a smaller cerebellum. You think about eye movements in PTSD, why does, eye movement therapies work for these cases? It's, again, an interesting thought. Just recently was able to present with Eric Moulton from Harvard. Again, a key role in this consensus paper on the role of cerebellum in perceptual processing. And then, and so you think about what we're about. We're about reading errors. We're about, in the vermis, we're about binocular function. We're about, again, PTSD and brain function. We're about perception. This is all connected with the cerebellum. Let's go and end with this test, dyskinesia test with cerebellar problem. So feet on the ground and ready to tap right to left. Ready, go. Go, 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 go. Make it go. Make it go. Make it hurt. Get those climbing fibers. Thank you. So next we're off to therapeutic eye movements. So why therapeutic eye movements? Because, and how are they connected to the cerebellum? And this is not to discount in any way all of the other connections in the brain. This is not a contest, but the cerebellum is so accessible. I'm hoping I can convince you so much a part of what we should be doing with the new research. And all eye movements require prediction, and sometimes with the head still, sometimes with vestibular head moving, often with both happening at the same time. Here's some very unique studies. You'll notice these are not done by optometrists, unfortunately. Does cerebellar function have to do with the feet? Saccadic eye movements? Yes. Does eye movements associated with multiple sclerosis sway? Yes. Can smooth eye movements affect pressure and muscle activity in one leg? Yes. Can decreased movements correlate with dynamic vision? Yes. Can posterior sway and cognitive workload be connected in young and old adults? Yes. Can the oculomotor task influence postural control in dyslexic children? Yes. Can you improve postural control in development? Yes, with saccades. Can smooth eye movements promote visual neglect correction? Yes. Can smooth eye movements help people with functional neglect with post-stroke? Yes. And here's a great randomized control study that just is not touted as much by Carrick and al. They used eye movement training after a mid-cerebellar artery stroke. In the control group, they went ahead and gave 3 control type of visual therapy 3 times a day, 10 minutes a day, using an iPad for 7 days. And the results were stunning. Everyone improved in the treatment group, and they had a significant increased change with just eye movements for 7 days. And eye movements are closely associated with eyes and ears. Excuse me. Here's the sound of ears. This is by Jennifer Grove's lab. Great book, Making Space. Great Coursera course we should all be taking. Making Space by Jennifer Grove. has a different sound. Eyes and ears are truly connected. So the cerebellar is a core function in the visual motor system to optimize performance. The cerebellum is intimately involved in real-time, immediate modulation of gaze shifting and gaze stabilization, as well as long-term calibration and adaptation of those skills. Intimately involved real-time change. And each canal has a different ability. We move our eyes to the right. That makes our canals to the right. We move our eyes to the left. We're going to go in more depth, but I just want you to understand the right cerebellum and left cerebellum, how they're interconnected with these canals. And our hero from eye muscles and against eye surgery is Jan. Small amounts of force are just required to move the eyes, and oculomotor therapy should target neural circuits. The cerebellum is renowned for its neuroplasticity, ability to learn, express procedural memories. Cerebellum with dysfunctions have been shown to work well with conventional musculoskeletal therapies there, and thus you should have therapies for oculomotor function and eye-hand coordination to achieve the best results. These multi-innervated muscle fibers that resist fatigue, they're predisposed to the oculomotor anomalies associated with smooth eye pursuits, gaze holding and gaze fixation, the flocculus, sensory inputs to increase neuroplasticity and adaptation, and they're liable for metabolic activity and must keep above a critical level to avoid detachment. Ocular exercise can serve attenuation in neuromuscular degeneration. Just all on point. Thank you, Jan. Let's end with dysmetria. If you don't mind putting your hands up and your pointer fingers out, and we're going to go ahead and back and forth and touching our nose, test our cerebellar dysmetria. Ready, go. Right, left. Go, no nosebleeds. Okay, end up.
Go.
Now we're on to deep definitions.